Hello students, welcome back to PhD Prep Lab. Today we will be discussing every important topics in biosafety and bioethics. These two topics are very important for ICMR and CSIR both. In biosafety, the topics that we will be covering are biorisk and assessment of risk, biohazard, WHO classification of risk groups, laboratory biosafety, and various biosafety levels. Those are BSL 1, 2, 3 and 4 level of labs. Complete notes and video PPT of this video will be also be available. Whereas in bioethics, the topics we will be discussing are Nuremberg Code 1947, ethical terms, ethical issues, ethical theories, ethical issues in research and human health. Mainly you have to focus on the ethical terms and ethical issues. Those are very important topics. So for this video, the PPT will also be available. So for the notes details, please stay tuned till the end of the video. Okay, so let's begin the video. So basically, what is biosafety and what is bioethics? Biosafety. In general, biosafety refers to the scientific practices, methods, and use of appropriate equipment in a biosafe environment to protect workers from laboratory-induced infections caused by handling disease-causing microorganisms. Whereas, in general, bioethics is the study of the social and moral responses that arise from scientific invention or experimentation, addressing ethical issues in life sciences by applying principles of morality and philosophy. If we talk about the history, the first report on laboratory associated infections was published at the beginning of the 12th century. However, 4,079 infections associated with the laboratory were reported with 168 deaths, somewhere between 1930 and 1978. So, the major agents responsible for these infections were Brucella species, dermatitis, Chlamydia piscati, Coccidiosis immitis, Coxella burnetii, hepatitis B virus, Salmonella typhi, Francisella tularensis, Mycobacterium tuberculosis, and Venezuela, and various encephalitis viruses. After this 1978 report, many more laboratory-associated infections and deaths were also reported. In these cases, also the infectious agents were same as Brucella species, Coxella burnetii, Mycobacterium shigella species, like this. When coming to the concept of assessment of biorisk, the process used for risk assessment are identification of the hazardous properties of a familiar infection agent or material, the activities responsible for pathogen exposure to the person, the chances of the exposure being a laboratory associated infection, the ultimate consequences of infection. Risk assessment needs very alert judgments. If risks are underestimated, they might result in serious and adverse consequences. Overestimation of risk can result in undue protective measures, which may be a burden for the laboratory. So, assessment of risk also needs to evaluate the factors responsible for infections, that is, whether it is due to hazardous agents or due to hazardous of laboratory procedure or working attitude of the laboratory staff. The staff's capability can be improved by appropriate training and including good laboratory practices with training of accidental spillage or inoculations. Now let's come to the topic of biohazards. Biohazards may be the microbial infections, infectious agent or other biological materials posing a risk for human health, parasites, viruses, prions, or biologically derived toxins, allergens, venoms, or recombinant DNA that can adversely affect human or animal health and environment. The properties of an agent which make it potentially hazardous are infection capability, ability to cause diseases in human or animal host, its minimum ineffective dose, severity of the disease, availability of vaccine, availability of therapeutic modality to control it, the probable route followed by it for transmission, stability in the environment, and host human interaction. The probable routes of pathogen transmission may be through exposed part of the body such as skin, eye or mucous membrane, through inhalation, through needle or sharp objects, through accidental ingestion, through bites of mosquito. By reading all these concepts, you can remember the 2020 breakout of COVID-19, which may be occurred due to these kind of miscellaneous things that the laboratory staff may have avoided the safety or something would have happened. Okay, now come to the next topic. The WHO, that is World Health Organization, has recommended the classification based upon the risk and route of transmission of the pathogenic agents in a laboratory environment, according to their disease causing capability and preventive measures available. The human pathogenic agents have been divided into 4 groups according to WHO and NIH, that is National Institute of Health. So, coming to risk group 1, the properties of microorganisms are microorganisms not cause any diseases, no history of disease in healthy adult human. Examples are non-pathogenic version of E. coli. Pathogenicity and ineffectivity of risk group 1 is it possesses minimal or individual community risk. Microorganisms does not cause disease in humans and animals. The preventability and treatment, that is preventative and therapeutic measures are available but usually not required. The biosafety level requirement of it is BSL 1. Coming to risk group 2, the properties of microorganisms in risk group 2 are disease causing microorganisms having low pathogenesis, disease is not very serious, preventive measures are present. Some of the examples of these risk 2 group microorganisms are measles and mumps virus. The pathogenicity and infectivity of risk group 2 are minimal individual and community risk. It is capable of causing diseases without serious symptoms. Sometimes it may lead to serious infection but manageable with treatment. Preventability and treatment options are preventive measures are available. Spread of infection is very limited. Biosafety level requirement to manage these kind of infections are BSL level 2. Coming to risk group 3, the properties of microorganisms in risk group 3 are these are virulent strains. The pathogen causes severe disease. The disease is aggressive. Therapeutic intervention is feasible. The worker is at high individual risk, like examples are hepatitis B. Pathogenicity and infectivity of these groups are high individual risk, pathogen capable of causing serious infection in humans or animals. Preventability and treatment, effective therapeutic interventions available, preventive measures available. Biosafety requirement of to or manage these kind of diseases are BSL 3 or BSL 4. Coming to risk group 4. Here the properties of microorganisms, so these are very high virulent strains, causes very serious disease which may be lethal, difficult to manage by therapeutic intervention. Vaccines are not available mostly. The pathogen possesses high individual and community risk like that of Ebola. Pathogenicity and infectivity of these risk group are it possesses high individual and community risk, highly contagious, causes serious infection in humans and animals. Effective therapeutic interventions are not available. Effective preventive measures are also not available. These kind of microorganisms are always managed in a biosafety laboratory of level 4. Now let's talk about, we have seen in these risk groups every microorganism is managed in a specific BSL, that is biosafety laboratory. So what is laboratory biosafety? National biosafety guidelines were established to ensure safe handling of microorganisms by recognizing hazards, assessing risk and implementing biocontainment measures. These include safe laboratory practices, protective equipment like gloves, mask, coat, etc., and structural features like containment barriers, sealed penetrations, specialized doors, and air handling system. Biosafety levels BSL 1 to 4 define increasing degrees of safety, with BSL 1 being the lowest and BSL 4 the highest. Based on pathogen properties such as infectivity, severity, contagiousness, and vaccine availability, the overall goal of biosafety practices and laboratory containment is to protect laboratory workers, the public, agriculture, and the environment from hazardous biological agents through proper engineering controls, protective measures, and safe work practices. So let us see one by one what are the different levels of BSL labs. Okay, so first BSL 1, Biosafety Laboratory Level 1. So here the agents are not pathogenic, well characterized, the risk level is minimum, facility requirements are standard laboratory is enough, Examples are we have discussed as earlier non-pathogenic version of E. coli and some yeast. The practices and safety equipment are standard microbiological practices are enough. PPE kits like gloves, lab coats, masks are enough. Coming to BSL2, the agents are pathogenic, risk level is moderate, facility requirements are limited access to other persons, biohazard signs are there available. Examples like the kind of microorganisms managed are mumps virus, salmonella species, etc. Coming to BSL3, these agents are indigenous, exotic, risk level is serious, facility requirements are controlled access, negative air pressure should be there, and example are coronavirus, West Nile virus, etc. And coming to BSL4, this is the most dangerous agents, and here the risk level is very high. These facility requirements are the facility should be isolated, full set protection should be there. The kind of microorganisms managed here are Ebola virus, variola, like this. Thus, briefly, biosafety requires effective policies for biosafety, biocontainment, and biosecurity, appropriate risk identification, assessment, and continuous monitoring for minimal chances of exposure. Training all those working in the laboratory regarding safety, responsibility, handling in accidental situations, and reporting of concern. Both workers and management should work with full cooperation and critically perform self review. Okay, until now we have discussed what is biosafety and different aspects of the biosafety. Now let us discuss about bioethics. So, coming to bioethics, biotechnology offers vast opportunities in medicine, agriculture, and sustainability, improving health, food security, and the environment. Its advancements, such as pesticide free crops, biofertilizers, renewable energy, and biodegradable products, have transformed human practices and thinking. However, manipulating genes in plants, animals, and microbes raises ethical concerns, giving rise to bioethics. So, rooted in medical ethics and moral philosophy, bioethics addresses questions of right and wrong in healthcare, genetics, and research. It extends from laboratories to industries and governments, ensuring responsible science. Research involving humans or animals requires approval from institutional research ethics or research ethics committees, which evaluate risk, benefit, and ethical implications. So, coming to the main aspect, uh, Nuremberg Code 1947. This code surfaced in the response of human rights abuses that had taken place under the Nazis during World War II. The war captives were subjected to abuses through experiments on human subjects. So this has took place during the World War II, where the Nazis, that is the Germans, have captured the captives, that is the slaves, and they were subjected to abuses through experiments on human subjects. This propounded the use of informal consent and non-maleficence, beneficence, justice, and autonomy. These provided normative framework used by research and medical practitioners. Now, let us discuss some most important ethical terms. Morality. It is a system of moral values and conduct which govern our decision of right and wrong or good and bad. Empathy. The ability of one to understand and share the feelings of the others. Euthanasia. It refers to painless killing of any terminally ill patient suffering from painful and incurable disease on demand of the patient and the court. Removing all life support system from a patient in irreversible coma is also termed as euthanasia. Autonomy. Autonomy is freedom from external control or influences. Independence. Justice. It is the treatment of behavior which is genuine, right, or just according to the prevailing laws. Equality. State of being at par or equal in status or ideology or opportunities. Beneficence. It is related to kindness, good, or charity for good. Non-maleficence. An act done to avoid harm or any act which would not harm or violate the trust of others. In the case of physicians, it is their act which will not harm the patient. And finally, accountability. The condition where somebody is held responsible, answerable, or accountable for an act. These all the terms are very important, and From these 10s only, last time ICMR 2024 questions have also come. You can also go and refer to the question paper of ICMR 2024, which is available in our Telegram group. So now let us come to the next topic of analyzing ethical issues. The ethical principles consist of certain virtues such as autonomy, non-maleficence, beneficence, and justice. Now the area has gained so much moment that before even small experiment work, one needs to justify its rightness or wrongness. Analyzing the ethical issues before starting any new experimental work would depend upon the assessment of these points. That is consequences. What would be the effect of this experiment? Who is going to be benefited? What would be the harms associated with this work? Thus weighing the possible outcomes and their efforts. Rights and responsibilities. Are we exploiting somebody else's rights? Do those rights need protection? Who would protect these? These are questions whose justification would have to be weighted. Autonomy. If one feels it is right, should things move on? Or else someone feels it is wrong, should things stop? Alternatively, who would decide things should be or should not be? These questions would again need to be addressed. Virtue ethics. Is this the best thing to do? What is good about it? Animal rights. One big question is, are we authorized to use animals the way we want, knowing that they too can think, they are aware of family members, they too feel pain at different levels, and they are alive. Animals like primates and whales resembling human brain features, family behavior, and some sensitive like humans should not be manipulated, but others should be. Why? Animals have been in use since long. They are used for manipulation of their genes as model system for study of human diseases and as transgenics for production of pharmaceuticals. Is inflicting pain in any form which may be creating a disease model or studying mutation in animals ethical or not? These things should be very importantly addressed before doing any kind of experiment, and these are still some kind of questions that are now coming to ethics and ethical theories. Environmental ethics. For everything, we are dependent on the environment. The environment and ecosystem are very nicely balanced. Thus, what would be the effect on the ecosystem once genetically modified organisms are introduced? What would the effect on biodiversity? Religious ethics. In this, the religious commands are being followed. It involves obeying divine commands and wills. Medical ethics. One of the oldest bioethics is medical ethics, which is in practice since its introduction as the Hippocratic Oath. The Hippocratic Oath is concerned about the behavior and relations of physicians with the patient, practicing non-maleficence and beneficence. However, medical ethics is not limited to the Hippocratic Oath. Philosophical ethics. In this, before executing any decision, it requires evaluation of reasoning and facts for ethical questions. Thus, all ethical decisions are taken upon evaluation of rational and logical questioning. Now, let us discuss some of the important ethical theories, that is, ethical egoism. This theory states that morality is mere fulfilling our self-interest. It criticizes the views associated with morality that are in the way of personal self-interest. It advocates that the actions are entirely dependent on desire and self-interest of the people. British philosopher Jeremy Bentham developed the utilization theory, which was later refined by John Stuart Mill. This theory advocates the selection of moral action which in its consequence brings greatest happiness for the large number of people. It works to decide the result or consequences of good action which would maximize happiness. Thus, it deals with consequences of action, in that happiness is judged because of some decision. However, greatest good for an individual or society, human race or animal is debated. Their supporters say that the theory aims to do good for the human race as a whole, including non-human animals. For example, terminal ill patients should be allowed for physicians assisted mercy killing to avoid pain and suffering. vegetarians also believe that abortion and infanticide may be possible in case the baby has several disabilities. Research on human embryo and genetic enhancement, which can benefit humans, may be made possible. Deontology. This theory, which states that morality arises from inner sense of duty, was influenced by the German philosopher Immanuel Kant. According to this, all people have rights and duties and they should follow these. It states that the decision should come from inner sense of duty without considering the consequences of action. Thus, according to Kant, any act that follows categorical imperative is moral. Kant theory requires the use of morality based on rationality with respect to persons and human dignity. Kant views refrain from hiding the truth from patients who have terminal illness. Kant categorically imperative. Act in such a way that you treat humanity, whether in your own person or in person of any other, never merely as a means to an end, but always at the same time as an end. It has been used to avoid abuses in research experiments on human subjects. Let us discuss some of the ethical issues on transgenic animals, genetically modified organisms and human health, stem cells, and also about human cloning. Transgenic animals. So, transgenic animal use is ethically controversial. We all know that. And modifications of any may cause sufferings and abnormalities to these animals. Strict ethical regulations guide their use, allowed only when no alternative exists. They should follow the 3R: replace, reduce, refund. To avoid these transgenic animal experiments, nowadays we can see that most of the experiments are conducted through computational model, so that we can avoid the killing of animals to study any of the, like, we are making disease models and killing animals. So to avoid that computational models, and lot has been developed in computational biology. Genetically modified organisms and human health. Genetically modified organisms may cause allergies and health risks. Seeds are costly and hurt small farmers. Lack of labeling lowers public trust. Genetically modified crops give higher yield and reduce pesticide use. They improve nutrition, shelf life, and make medicines. And the next one is stem cells. Stem cells can differentiate into many cell types and may cure several diseases. In the future, they could help generate whole organs through tissue engineering. Types: adult stem cells repair tissues, embryonic stem cells form blastocyst, cord blood stem cells form umbilical cord. Ethical issues: using and destroying embryos for embryonic stem cells is seen as immoral. Adult and cord blood stems are ethical alternatives, but rejection and uncontrolled growth remain challenges. So, a lot of research is going in stem cell area to use in every aspect. Next is human cloning. Human cloning can be reproductive or therapeutic using SCNT. Supporters believe cloning may help treat infertility and advanced therapies. Opponents object to embryo destruction and risk to egg donors. Most countries ban reproductive cloning as unethical and against human dignity. UNESCO, WHO, and GU, that is Europe countries' declarations prohibit human reproductive cloning, stressing caution in its use. Detailed notes and PPT PDF of this video are also available. So to get that, click the link in the description, or you can also follow us in our Telegram channel. The Telegram channel link is also mentioned in the description. For more updates and subject related content, please do follow, like, share and subscribe our channel. So until next video, please stay tuned. And for the next video, we are going to bring the MCQ practice for these two topics, which are very, very important. So stay tuned for that.